you're audible. Yes.
Okay, so hello everyone. Thank you so much for joining here this afternoon. I'm Masters in Nuclear Medicine from IIT Kharagpur and Tata Medicine in Kolkata, and in DDMRC Caritas Hospital as a nuclear medicine physicist. So today we are glad that we have Dr. N. Ramamoorthy with us. So he is an eminent radiochemist and radiopharmaceutical scientist by profession, and he attended BARC Nuclear Training School, and he did his PhD in chemistry from University of Bombay. And presently he is a senior advisor to director of BARC and adjunct professor of NIS Bangalore. So apart from these, he was appointed in various positions throughout his career, which includes like he was the visiting professor at University of Missouri, Columbia, at USA. He was the head of radiopharmaceutical division at BRC, and also the head of RP radiopharmaceutical program at BRIT, and also chief executive of BRIT and associate director of isotope group and at BRC. He was also the director of the division of physical and chemical sciences at IAEA, Austria. So he has 40 years of professional experience in the field of radioisotopes and radiopharmaceutical development, production, and utilization, and many more things. And apart from his awards, he received multiple national and international awards. So if go and trace back like how NM started in India, so and especially the history of PET in India. So he was the first person, like when RMC procured the first cyclotron, he was the first person after the first batch of FDG production, the first person on whom the FDG was being injected and the imaging of the PET CT was being done. And now we can see how globally PET is an universal imaging modality for cancer diagnosis, detection, and many more things. So now over to Joshi for the next proceedings. Thank you.
Thank you, Biprojit. Thanks a lot for introducing, sir. And I would like to make a personal note on this. So we have been, because I haven't been much there in nuclear medicine, so it's been 3 or 4 years only. But I have been to a few conferences, and whenever we were there in that conference, we could see the intensity or the path sir has led in the radiopharmaceutical chemistry. So we would like to take those cues from you, sir, and we would like to proceed on further with those things. So shall we start with that, sir? Before I would like to start this, I ask Dr. Ramamoorthy, sir, to say a few words to address the gathering.
Okay. Thank you, Rakesh and Biprojit. I think it's a good initiative, and I'm happy that you people contacted me and then asked me to join for an interactive session. So I was really looking forward to it. And when we were just looking at the dates and when you people suggested, I didn't realize the nice coincidence. As you all know, today is the National Science Day. I think that's a very good, noble, kind of a woman that you people started on something, and it actually coincided with a very important day of the National Science. This was, of course, not by design, purely out of convenience. We put a date, and it turned out to be like good. So I'm pleased to know that Kharagpur IIT type is running a kind of the nuclear medicine program, and some of you have graduated, and there are more students who are interested in the whole thing. So I think it's a very important, good field. And we would definitely, try to see like how best. Obviously, in a limited time of 40-45 minutes, we cannot be doing, far too many things. But as we were just, initially telling, I was thinking that I'm going to be talking to really quite young people who are probably keen to know, have I done the right thing? Is there a good future? And what do I do? Whether to, be in research or be in, business or what it is like. Those kind of questions also probably are likely to be more than perhaps only the technical. So that's what I was, so I'm sort of envisaging, but I really do not know like the depth and breadth of what has been taught to you people during your actual course. I have not bothered to really, very frankly, to look up like what's been the syllabus like or what you people have all learned like. Let's hope that that would be a productive, good session and perhaps a good beginning. And we will, I would like to go whatever way the organizers have planned and done it, like, even though I was initially expecting kind of a broad-based. I think we are all cricketers. Most of you love to know about cricket. So I think I want to just give an analogy. It looked to me like when Rakesh first said, we are going to discuss our Indian cricket. So I was very happy. Okay, we are going to talk about a lot about Indian cricket. Then he suddenly says, sir, we are going to talk Akshar Patel and Ashwin. They are very important, true, very nice. But that's alone is not the Indian cricket. As you know, it's very vast. one either can talk only about the bowling or the batting. Even that would be a vast subject. So that's just to put things in a lighter vein, to make it more informal for all of you to feel free. Not that all the questions I will know the answer, but certainly, we would try to respond, or, try to send subsequently emails. My email ID contacts are available with these people. So I think that there I would stop and greet you all, and then let's go ahead with the program, whatever way it's planned. Thank you. Over back to you people.
Yes, sir. Sure, sir. Even we have planned with Actinium also the broad perspectives, the recurring themes, sir. So we will surely like to take those things only. So I'll proceed on with first few questions, and I would request all the audience to kindly post your questions in the chat box so that we can take it up as soon as possible. So anything, and I would like to make a request also. Sir is ready for all the questions, and since we are at student level, so I would like not to hesitate for posting any questions. Everything will be taken up here. So I would like to make that request, and straight away I'm going to start with Actinium Radiopharmaceuticals. Is my screen visible, Biprojit?
Yes, visible.
First question we would like to initiate is among the we have various alpha emitters that are there. I mean, like it's a group of radionuclides. So why is it that Actinium-225 is gaining more importance, or is more preferred nowadays by the scientists or the clinicians?
Rakesh, would there be similar questions, or these are already like consolidated, therefore you would like to begin with that?
Yes, sir. These are consolidated only, sir. The minimum questions. Yes, sir.
Okay. I think, let's then take it in that same manner. Like, see, this actually, takes you back to some basic principles about what you call as actually the radioisotope based, or the radionuclide therapy as it is called. Your intention is what? You want to get some radiation to a certain cells or an unwanted area and get them completely, irradiated and destroyed. So it's best done if a particular isotope is such that it exactly puts all its radiation right on that lesion or that troubled spot alone and not anywhere else. In such a kind of consideration, the alphas score well because they are going to have a very limited area of where the radiation will be deposited. And that was the interest. This is something like very well known. Therefore, everybody was interested in alpha. But then you need to have some actual practical situation where it could be like, used, and a kind of, what would be an appropriate one. Like, I think the very first success came with the radium-223 for the bone pain palliation part. You see, the bone pain palliation itself has got a long history. Everybody knows that the alkaline earth metals go to the bones. So you have a strontium-89 beta emitter isotope. And in the case of cancer patients, many of the cancers, the spread is in the bones. And ultimately, it leads to a lot of pain, and quality of life comes down. They are terminal patients. They are not going to be cured. Purely a palliative treatment was possible using those kind of the bone seeker radiopharmaceuticals. So the bone seeker radiopharmaceuticals in strontium work. The idea of the radium occurring in the same period and an alpha emitter, it could really be sort of, very good. That in fact was the real starting point with reference to the alphas being used. And obviously in the very early stage, there was a lot of excitement in that. But as I mentioned, that's not a curative one. It's palliative. Those people are terminal patients. They are going to meet their end sooner or later. And it's an improvement of the quality of life till their survival time. That's what is being to be achieved by the nuclear medicine procedure and with the isotope. And therefore came the radium part of it. And since it did some nice wonders, very good results, in the particular case of out of the few cancers which spread to the bone, prostate cancer is also one of them. And since there they found certain real good results, and that prompted to see like maybe we should try with some other alphas also with reference to the prostate cancer. There, obviously, amongst the other things in terms of the half-life, the bismuth 213.46 minutes was available, and its parent nuclide in the chain is nothing but what is actinium-225. In fact, the actinium-225, as the slide you have projected shows the series of decays, it comes down to that bismuth 213 part. So instead of struggling with a less than 1 hour half-life isotope, if this sequential decay could itself be sustained, why not directly try with the actinium, because it gives you the advantage of the 10-day half-life also. Like, I think this is my feeling of the genesis. I have no absolute proof exactly this was the genesis with which it said, but I think it would survive quite some amount of a discussion, arguments, and that's how the actinium actually came important into the picture. I would stop there because just on one question we can go along, all the 45 minutes will be over.
Yes.
But that's not the intention here. I'll stop there.
Yes, sir. Next question, I'll move on further. So this is again the continuation, sir. So what limitations does actinium-225 have in terms of wide availability and effectiveness, and which production method in India we can see being coming up?
Yeah, it's a good important question, very practical one. Like, I do not know like how many of you attended the Nuclear Medicine Physicists Association conference about 2 weeks back. There, a little bit provocatively, I have talked about this topic because its availability today worldwide is extremely limited. What's available today will be hardly sufficient for few thousand patients, whereas what they are expecting is somewhere upwards of 500 times that kind of a requirement. That's the type of the larger number of requirement which is there. Like, so naturally, the international corporate world as well as the research technology scientists are looking at, and the main methods are going to be coming through from the thorium, what's called as the spallation reaction. Like, essentially, it means you have very high energy protons which produce too much of turmoil, throws out large number of neutrons and protons. By the very word spallation, obviously the end product is not a single product. There are going to be a series of product nuclides formed, out of which your interest is in actinium-225. And that's the large, it can produce large quantities. Therefore, that's what is being concentrated by the global corporates along with major technological institutes in US and Canada. And that procedure is going to probably give eventually the largest amounts. Like, but that naturally needs a high energy proton accelerator machine, the type of which we do not have currently in India. Once the importance of actinium-225 had been known, you take the periodic table, you take the nuclide chart and start playing around with what are all the other possible reactions. 2 more reactions are possible. One of them is listed in your own listing there, like radium-226 proton-induced reaction, throwing out 2 neutrons, which gives rise to actinium-225 directly. This reaction is something possible at about 25 MeV energy protons, and there are many cyclotrons. You know that in the one of the past we had a very important isotope thallium-201, to produce which you needed 30 MeV cyclotrons. So they have been there, and a good company IBA has very robust machines. Such machines can theoretically produce. But what are the kind of difficulties, technologies? Radium-226 is something which everybody would like to keep away from. It's a messy radioactive material. And to have that as a target and have a very prolonged irradiation where you are going to necessarily bombard with a lot of energy intensity, and therefore there is going to be tremendous amount of heat generated. That's a very, very tough, challenging targetry development, but that's worth it because it's going to give you actinium-225. Therefore, this P2N route is possibly in India also feasible because we have such a cyclotron recently commissioned about a year back, though with much delay. The facility is finally on and available at Kolkata, and therefore that could be. But having said that, in the same breath, it's not like something like tomorrow or day after. It's an extremely involved. First of all, the target radium-226 will not be there. There was long back a time when a lot of such radium was all scooped up and taken as waste into disposal arrangement because it was hazardous material. And now today we are saying that would be an interesting target. Can we somehow recover it? So perhaps people are looking at it, and radium-226 when it comes through and you have a safe, good target, the scientists, technologists have capabilities. It will have to be a concerted effort. It's not going to take place over days or weeks or months. But if there is a concerted effort, internationally also it's going on. Somebody hits a jackpot, comes out with an excellent, good target, perhaps that's going to be a very expensive, or obviously the intellectual property part will be there. But for younger elements, working in collaboration, particularly institutes like IIT Kharagpur, where perhaps we have excellent engineering, or not too far away from Kolkata VECC, perhaps an excellent developmental initiative. It could be a big jackpot, but it's by no means easy. I want to underline, emphasize that. But if something could be done, that's great. That will be one methodology. Like the second one is the same radium-226 target, and you can go through with a photonuclear reaction. That means gamma photons are actually the impinging instead of the particle. It is the gammas, and then the gamma in reaction on radium-226 will give rise to radium-225, which decays with a 14 days half-life or so into actinium-225. So if there is an electron accelerator of about 25 to 30 MeV electron acceleration, that converted in the Bremsstrahlung effect, and you will be able to convert, and then that energies can be used for the photonuclear reaction to produce. This is something which could not have been very readily, quickly thought of, but happened to have, having had the interaction in the past with so many years of Indian experience, what I recall and remember is there is an institute called the SAMIR. That's actually something for microelectronic thing. It comes under the Department of Electronics, which is an autonomous body, SAMIR. And they have about 10 years back got some grant and money to address the issue of molybdenum production. It's in another isotope, which many of you will know, like using a similar type of a photonuclear reaction route. There had been some delays in terms of perhaps funds and other availability, but the project, I believe, is still on. So therefore, when that machine comes up, that's not only for only the molybdenum production, where with molybdenum 100 target gamma N producing you molybdenum 99, you can also use that same type of a machine, provided this would be possible. Purely at an informal level, some project people whom I know, I have taken the liberty of telling them, I did articulate this view in that NMPA conference, and that I mentioned to them, sent my slides also like. But again, that's not something like a secret or a closed one. It's a Government of India national resource used project. Perhaps in future, that could also be. Again, the same radium-226 target. The targetry, technology issues, challenges will be there. They cannot be wished away. But you have Cyclone 30 at VECC Calcutta. Samir is building up an electron accelerator of somewhere around 25 to 30 MeV electron accelerator, which is also meant for exploring molybdenum production. So it could also explore this. Perhaps, one will have to take it up with the right people in the institute in a very formal way. But purely, giving an idea is something relatively simple. I have a few slides and articulation, so I talk, and that's not going to produce you actinium-225. Lots more groundwork, on the field work will have to be done. These are the two which I would consider as going to give you reasonable quantity for a long time. But there may be some very old stock of thorium-229 in the BARC, which our colleagues there have been trying to locate and take. That could give perhaps in the near future some very small quantities, because it's more than a 7,000 and odd year half-life isotope and very limited, and I don't think there is so much of legacy waste there. But whatever today internationally you are getting is coming from such legacy waste accumulated in the past for some other research also. So if that legacy waste equivalent, some small quantity which our BRC colleagues are trying to gather, if it's coming out, there may be some small quantities available from there. But again, that will be relatively very small quantity. For a long term, both the Cyclone 30 and the Samir's electron accelerator to come, the authorities will have to be told the importance of actinium. Therefore, this could be built in. Thanks. I'll stop there.
Yes, sir. Next question. Yes, sir. This is again, given the situation of wide and cost-effective availability of actinium-225, is it versatile in terms of chemistry to produce variety of radiopharmaceutical? Because we are aiming at different diseases. So is it, versatile with respect to its chemistry? And continuation of that question. So what about the in vivo stability of daughter radiopharmaceutical? As you have already mentioned, like it decays to daughters which are also radioactive. So, and there is a recoil effect associated with alpha particle decay. So there is always a possibility that they can get lodged out of the radiopharmaceutical. With the pharmaceutical, they can get disintegrated. So how is it going to be?
Good, good thinking and good question. Like, let's put it this way. Actinium today, we are not treating or exactly equating it to say iodine-131. Iodine-131 has been able to go as a therapeutic nuclide with many, many things like. That's not necessarily the case. Even if you have one or two vectors with which some very important lesions are going to be treated, that's going to be an adequately satisfactory situation. You don't need to do so many things with say the same one isotope. Like that's in fact the beauty of nuclear medicine. You have a huge varieties available, and for whichever vector, whatever could be a right isotope, which could element chemistry wise and energies wise will fit, you can always use it. Theoretically, yes, recoil effect and some small break-in, and because of that, certain additional, for example, the francium is equivalent to your, alkali metal ones. So a small quantity could be there even going into the myocardium. Those things have been considered, and at least as of now, what the literature says wise, there are points to definitely be kept in mind. The students and the practicing people should know about it. But I don't think any of them have right now come into any situation where that's going to be a criticality. But always in the therapeutic products, what is the criticality is, other than the target organ, there would be some other tissues also where the material goes. That probably is the greater limitation with reference to many of the therapeutic radiopharmaceuticals, including the actinium one. Like the in a recoil effect part would be definitely be there, but as of now, with the few type of vectors, whatever they are trying, like I think, and particularly concentrating one major thing, that is the prostate cancer, where the prevalence and the incidence things are very high. If that's going to be sort of served, like it's good enough. It need not be doing like, as I said, everything need not be met by only one particular isotope, and we have not gone to that. That's why, in fact, on a jocular vein, like I was picking up on the cricket analogy, the other one I had is, you people are quite optimistic because you have used the word plural, radiopharmaceuticals. I would have said we are probably still with only in actinium radiopharmaceutical, only one of them. That's the actinium PSMA ligand conjugate. That's currently the only one which has seen the light of the day. Like there could be many others which are being explored in the laboratories, but I'm not really too sure how would it be like, particularly anything which could be taking a very longer time to reach the targets. There could be a bit of a problem with this kind of a decay chain isotopes. I'll stop there.
Yes. So the next question, sir. One sec. Yes, sir. This is again a comparison. Yes, sir. So since targeted alpha therapy removes efficacious or lethal in terms of radiation damaging, are we moving from lutetium or beta emitters era to actinium or alpha emitters era? And the next part of this question is alpha particle therapy the fundamental limit of radionuclide therapy? That means, can we not go beyond alpha particle therapy in terms of radiation damage with nuclear medicine?
I think, part of this, we already in my kind of initial remark, part of it I covered by telling you what's theoretically known and because of which you aspire for that versus what is practically doable and what you try to do. With reference to any of these kind of alphas or alpha equivalent to your internal conversion electron-based therapy also, if you take the internalization, that is the particular radionuclide along with the vector really getting internalized, becomes actually the limiting arrangement with reference to availing its therapy, which I believe has been fairly a big challenge. And that is how the very generic ones, where it could be a little bit more penetrative, but doesn't matter. That's how much of the beta emitters have scored. I would not believe that we would be moving away from lutetium into alphas in any near future. It all takes long time. Lutetium has a very good physical properties. Its beta energy is not extremely large, therefore its radiation zone where the deposition of energy takes place is confined. So lutetium is still extremely important. It would be doing it. With reference to the kind of fundamental limit, like what we say is, more than the radionuclide, how much you can ensure that you are going to have internalization of your actual product molecules that you are taking. The radioactivity as well as the vector together, that radioactive radiopharmaceutical molecules which you are taking, how much you can get it internalized in the cells which you want to destroy. That's going to be the fundamental limit. That's what I would put it in a broader base. I stop there.
We have one question from the audience, sir. Yes, thank you, sir. We have one question from the audience for both primary and advanced stage.
The chat box, I can open it. Yeah, for both primary and advanced prostate, is actinium PSMA a good choice? I would say it should be put to a clinician probably, but what we today know is
Yes,
like
sir, yes.
the castration resistant cases, which is supposed to be a large percentage of the prostate cancer people, and that is where it is. And this so-called advanced stage, what has currently happened is that people who have gone through a huge amount of treatment and there had been a lot of problems, nothing had worked like, they are ready to try anything new. And that's how most of the cases which have come are being handled very initially in the so-called, for that matter, even the lutetium PSMA therapy or subsequently actinium PSMA therapy has been these kind of very advanced cases. Like, as the results are seen, and if the nuclear medicine is able to win over the other referral medical community, and it could enter as what we call standard of care, that standard of care entry, if it takes place, probably people will be coming even earlier. The efficacy will be much, much higher when that kind of a stage comes on. But as of to begin with, whatever had happened is to do with the people who are actually given up like by the other methodology type. So therefore you have got it like coming. So I don't think like one should really look at kind of an extremely advanced stage. It need not go up to that stage. Like, your procedure is compelling and the results are very well proven, it could enter as a standard of care a little earlier. When that takes place, probably you would find a much higher value contributed by the nuclear medicine than what we are now showing as virtually towards the end, like last bit come up. Yeah, I think with the lutetium, we already have people started recognizing and accepting and bringing the referrals much earlier on. And I hope the same thing will continue, and also you would be able to convince and get that procedure getting under the standard of care routing. It's not easy. It's not in a day or so done. Lots and lots will have to be done by both the nuclear medicine physicians and the referrals to really accept that kind of a status. Thank you.
Yes, sir. Thank you, sir. We'll move on to the next question from our side. So that we have therapy or these over. So far this has been the case. So I'd like to make an insightful thing, or like maybe an a small thought. So since cancer cells are better characterized by rapid mutation rate, so it's as we at the end of the everything else has failed and we are dealing it with radionuclide therapy, and after a while we see that that too fails, and then there is no other choice. So are attempts are there or molecules to target that rapid mutation rates? I mean, like if you have some molecule responsible for rapid mutation rates, so that we can have sustained radionuclide therapy, because since our target is now at the fundamental level.
I think, you have a very valid point there. Yes, you need to have a starting point. That was the very idea of everything on these lesions are being, whether it is for imaging, then comes the therapy. The lesion, even targeting the lesion, you got to have some kind of a signal to be able to first to do it. So, on day one, if everything will be a variable parameter, you would not know like where to start or where. Now that it's been known, what could be the kind of the signals with which you can start working on, like as the next generation thing, like in terms of the R&D level? Yes, very much. Your point is extremely valid. So certainly, the R&D can be looking at it, but that should be like a kind of an evolution and not challenging what is going on. Because, you need initially some form of an arrangement with which there is a signal there in the lesion, which I can try to log on to, and then I will be able to hold and get an image done or a therapy possibility. Now, that being done, that should be supported by all means. Then you also give into the point that this could continually be changing. So I need to be looking at it like this is an absolute evolution process. All of us remember, particularly some of the, I don't, most of you are younger ones like which you may not. You started with a 50cc moped TVS 50. That was the kind of the two-wheeler. And then now virtually you have a 800cc, more than sometimes of a car level, power level in a two-wheeler itself. Like it's a process of evolution. The 50cc TVS 50 has had its role. Nothing wrong with that. Like the same way here, you needed some kind of a spot with which you can target on a lesion, and there whatever would be a very good one, be it an overexpressed receptor or there is some other thing with which you can go and hatch on to, or you can do some other kind of chemical mimics, etc., etc. All of them have worked, but you got to be receptive that this may not be the ultimate. We may need to have an additionality. Let that please come in the process of evolution. That should not start telling like there is a deficiency in the current approach. The current approach is extremely valid and sound, and that's what's given you an entry into the field and your ability to contribute to medical management of patients and then be able to be there in the nuclear medicine. There is always scope for improvement, and in that process, some of these new ones will come. For example, if you take the antibodies were the ones which were much more earlier kind of, fancied as the right target, vectors to carry. Like it didn't work, then came down to the peptides, and then instead of, thing like you looked at, receptor and receptor binding peptides became a more easy one to be able to do it. Like, there is always a continuous evolution. This I would like to emphasize. Thank you.
Yes, sir. So I would request audience to post your question. So our host has exhausted question. I mean, like we have come to these questions. So I would like to ask you on the research reactor that has been approved for this public-private partnership model. So I would like to ask you your comments on that, sir.
That's something, which I would know only whatever is there on the website of the DAE and BARC, and wherever chairman goes, I think he has been talking about it, and whatever you are seeing, like it's a kind of a very, bold move by the government. And if there are stakeholders, how much are they willing to come in with their resources to partner and do? It's exploratory. I'm not saying that tomorrow it's going to exactly work. How much will have to be government money? How much will have to be the private people resources? We do not know. Like that's in fact the reason why in my talk in that nuclear medicine physics group, I provoked, instead of such a big thing like a research reactor, why not simply an enriched target production program in a public-private partnership mode? Like maybe there are some good entrepreneurs with science and technology based people available, and making an enrichment facility thing need not really look that great a difficulty like a research reactor running. Still there will be resources involved, but probably one can look at it like more like drawing an analogy from the government's move. I provoked with that question. Like I'm not expecting somebody will jump into it tomorrow, but the point was how much the stakeholders are really keen or willing to come in with some resources to be able to partner. But always there can be a worry, with government, if we go tomorrow, if everything is taken away by government, what do I do? Those kind of things they will have to work out. Like it is in the very early stage. It's a kind of a innovative, good thinking. What would be their plan, etc., which we would not know. That would be known only within the government and the Department of Atomic Energy. But plenty of information is available, and I think they have engaged a consultant. That's what the media web says, and people could be in touch with that website to be able to get update information on that. Thank you.
Yes, sir. So we have one more question from the audience. So what about ActiniumFap? If it's possible, then we have a solution for wide variety of cancer treatment. So I would, I would like to add my comments also, sir, on your comments on FAPI first.
Biprojit, I am not really literature savvy to fully know about the status of currently the FAP. But anything which is sort of telling like that, some things could work for a wide variety, I will take it with a pinch of salt or a pound of salt, because we have had problem in the past. So I do not want to jump or generalize to say like that it could. If it works well and good, no problem, but not so sure. Rakesh, you go ahead with your comment.
Yes, sir. How do you see fibroblast, that same enzyme inhibitor? So how do we see its impact in future going to be on nuclear medicine with respect to diagnostic imaging?
Yeah, again, like, I mean, my current information with reference to the literature, recent ones, is really not that very deep for me to be able to make a comment. But yes, you referred to the diagnostic imaging part rather than the therapy part of it. Like, yes, in the diagnostic imaging, like even with certain degree of lower specificity problem, it is still can have a very good impact. So that way, instead of an FDG based, where we are doing so many things with one FDG is like a, kind of a lesion marker for many kind of a situation. There could be a scope, means yes, that kind of a imaging part, wider applicability is more conceivable rather than for therapy. I would leave it at that.
Yes, sir. There is one more question from the audience. Sir, can you please comment on the abscopal effect in alpha targeted radionuclide therapy?
Priya Sharma, can you tell me what you mean by abscopal effect? I am not really able to comprehend that word.
Audible?
Yeah, you are audible. Yeah.
Yes, you are audible.
So, abscopal effect, I mean, no, not directly targeting, but targeting the cells by alpha therapy directly. The cells which are not
Voice is
in
breaking.
the
Maybe
target
somebody
site
else
are
who heard
also
it may repeat
getting.
this
Yes,
statement. I couldn't get the
Yes,
words fully.
you
Somebody else had heard it fully. They may please rephrase the question, please.
if you have difficulty, Priya Sharma, you can type it. We can read it from there also, because your voice is not so clear. Yes, she'll take some time.
We can go ahead with other questions. Let that word chat box come and we will see that.
Sir, basically by abscopal, they mean that the untreated tumor cells, they also shrink. Sir, am I audible?
Yeah.
Sir, by abscopal, she means that the tumor cells which are not treated by the radiation directly, they also shrink.
Okay. I mean, this I would really leave it more to the radiation biologist to be able to more comprehensively answer. I would be rather too superficial with my knowledge to respond to this question. Sorry.
Thank you, sir. So I would request audience also to speak up if there are any questions. So I would proceed on with the host discussion. Sir, what are the opportunities available for R&D, research and development in terms of institutions in India for for people who are going to are interested in a career in radiopharmaceutical chemistry?
This is something which I definitely expected will come up. The good point today is long back, the nuclear medicine or radiopharmaceutical anything means it was all only confines within the confines of Department of Atomic Energy. Whereas today, all the major medical centers where there is a flourishing good nuclear medicine unit, there are excellent scientific people working there. You take AIMS, you take PGI, SGPIMS, Christian Medical College, Vellore. I'm just picking few. It's not that they are my favorites. There are many, and there are good cases. This has also come about because of the medical cyclotrons becoming popular and they're being operated in many of the centers. So they do give a chance. Only thing is, what I'm not too sure is like how much the institutions would like to exclusively specialize for a scientific radiopharmaceutical angle alone. They may probably prefer a broad-based nuclear medicine person who are probably handling imaging systems also, and they are also operating the cyclotrons, those kind of a thing. So in terms of this type of a situation, how much will be purely for researchers, there would be scope is difficult. But suppose there are fellowships available, these are few institute names which I took. INMAS, I forgot to mention, Institute of Nuclear Medicine and Allied Sciences. They are all the places where probably people could be there. And again, today, the word, it's not never nation is in that narrow sense, like you have regional as well as global opportunities and possibilities. The field is a growing field, and as many of you will know, plenty of our India-based people, be it in science side or a medical side, have been doing extremely well in nuclear medicine field all over. And many of the foreign universities, professors, do have active radiopharmaceutical program. They won't have necessarily an active nuclear medicine program, but they have an active radiopharmaceutical development, program, science-based program. Like there all could be opportunities for some of the people. One more thing I would say also is the entrepreneurship. Like, we have had the nuclear medicine technology course from which many have come out. Quite a large number of them are doing excellent work in the nuclear medicine departments, but many have moved over to the industry, corporate side itself also. And some have probably started their own entrepreneurship. Why not that for the nuclear medicine science person also? So I mentioned, you all know, like there are over 1,200 cyclotrons. You look up whether enriched target international market is already kind of saturated or not. And if you think there is a scope, why not an industry for the enriched targets? Make in India. Our government had been extremely keen. So you can have a make in India type of a thing. So there can be entrepreneurship kind of a scope also. I don't want to sort of, undermine that, but I'm only telling that one can have multiple. It can be in a very regular clinical management, clinical support services. It could be in R&D. It could be in entrepreneurship. That's what I would leave it at that.
Yes, sir. So the next question. So among all the radiopharmaceutical vectors, I mean the pharmaceutical parts are the localizing agent. So which one you think will be better for diagnostic imaging? Because nowadays there has been a lot of things like we have peptides, we have small molecule inhibitors, as well as these enzyme inhibitors and antibodies are also coming up. So among all these things, I mean, there are various they are variable on several grounds, but which among them you think will sustain for diagnostic imaging?
I would approach it from the other way, because you are talking to a near 70-year-old person. So we have learned it the hard way. So you people need not learn it the hard way. You start from what is needed. That's my current advice. One can say becoming wiser after all the events, but it's probably that. What is it, that single thing which would score in the medical world? And that, even if it's a jackpot, it's very difficult to achieve. If that could be done by a radiopharmaceutical and a nuclear medicine procedure, we will be in great business. Rather than seeing what all we can do and then trying to find out an application for it. So in that sense, where all now we are making a bigger impact in medical imaging. If you take such kind of areas, then in that, what are the current agents, what is the deficiency, and what could be done? Then you come around to deciding whether I should be with an extremely small molecule or modest size, or even a larger molecular weight material does not matter. Because there your end goal is fixed. I need to be able to target and be able to do it, say, within this 1 hour. Within 1 hour, it should get targeted. It should remain for 6 hours or so. You are defining what would be like a wishful list for an ideal imaging thing. So that once it is done, then you come to decide what will have to be the right carrier molecule. And one good thing what has happened is, it's not necessary everything has to be done only by SPECT or everything has to be done only by PET. Both are well accepted, both in both the instrumentation system, excellent advances are there, and therefore there is very good resolution, high quality imaging, quantitation, availability, all are there. Therefore, the nuclide wise it can come. What will have to be my target lesion for which I have to do the imaging, and which would be then my best vector, and then which isotope is best to match with that vector. So going backwards, taking a clinical problem which is currently having a deficiency in terms of any other alternates which is being used would really be the ideal. Or there are agents which are doing, but we could do it better. For example, that would be the example for the myocardial perfusion imaging. We have been doing the myocardial perfusion imaging earlier with thallium. Then we had the technetium cationic lipophilic complexes doing it. And now you can do it with ammonia or rubidium-82. Or you are looking at the other angle, the viability directly using the FDG also. But still, myocardial perfusion imaging, quantitation, ability to do with PET is good. So probably one important agent would be something which will have all the properties of thallium, but which is doable with a PET-based pharmaceutical. That would probably be a big achievement. It's a great milestone. It's not there today, but people have tried for it. Like there are some products with F18 also done. Like somebody would ask if there would be something like being made that could probably make a big difference in the nuclear cardiology practice, because the quantitation ability, the high quality, superior resolution images, what is possible with the PET-CT thing, like could probably. So try to approach it from the other side. Do not, once you, of course, you are on a particular type of a product, then in that given situation, you have to see whether your best vector molecule is a very small molecule or a larger molecule. But ideal will be to come back from a clinical question. What would be today really hurting? And that if you get, and then you feel something could be done. That's of course quite a bit of a challenging thing, but it's possible to do. For example, we all know about the infection imaging, and that people knew it would be of help, and therefore it sort of started off, and then we had agents which are not so very satisfactory. Then we had many other things, including FDG today, what is used for that kind of a thing could be done. But leukocytes, WBC labeling became a landmark thing, because like there, if you are doing an infection imaging, it's going to be helpful. That's the kind of the clinical. So I would emphasize on the clinical end goal being something very clear and fixed, which would really make a driving difference. Because for routine things, what we can do in nuclear medicine, we already have agents. Something is going on. So you would like to try, I would advise very seriously, start with where is it lacking? What is it that we can really add as a high value contribution? And that's, I know it's a little bit more ambitious, but since you are talking on the National Science Day, I had already planned this question will come and I will give this answer. Thank you. Thanks for giving that question opportunity.
Yes, sir. Sir, one last question for the session we would like to take. So, if we have a radiopharmaceutical vector and we have a radionuclide that comes out to be a radiometal, what hurdles do you see to make it into a radiopharmaceutical? So, radiometal with a given vector. So, what are the important things in its labeling?
Yeah, that's where finally that word, whatever we have used in the title, till now what we talked about was broad radiopharmaceutical science.
Yes.
That's where the word chemistry comes. Chemists are, ingenious. You need to ensure that you are able to put the radionuclide with the vector without or with least disturbance to the vector property to bind. That's the crux of the matter. This is very easily said in one sentence or one slide. To actually achieve it in the lab is really the challenge. How much we are able to retain the vector properties without being kind of vitiated by doing that attachment in between moiety to link it and then bring it to that. Of course, there are plenty of success stories available are there, but that doesn't mean that same chain will work for yet another nuclide or yet another vector. That's what gives rise to a lot of research scope for you people. So depending on the particular vector and which is its active moiety which binds, and then which is actually the end which you can tinker around to link with the radionuclide. This is where it comes to a lot of amount of trial and error. Perhaps computational chemistry can be an extremely helpful thing, like knowing clearly well which is the important moiety of the vector which should be protected and which is something okay to sort of little bit play around, and then your radionuclide, radiometal getting linked to that in that type of a thing. That's where a lot of chemistry research could be there, and I am pretty sure that's how institutes like your IITs or other academic organizations and the nuclear medical centers like coming together, including in India. This is probably there in many other foreign countries, but in our end probably that's not there, and that would probably be a good way. Let's stop it there on that happy, positive note. Thank you.
Yes, sir. Thank you, sir. Thanks a lot once again for initially to having accepted our invitation to come up. I would like to conclude on one mark. So this has always been our motivation. So if I have seen further than others, it is by standing up on the shoulders of giants. So you all have been doing this radiopharmaceutical chemistry, and we know how much crucial you have been to develop radiopharmaceutical, that too in would like to stand on your shoulders and take it as it is with the same motivation and say. And I would like to ask you, thank you once again, and I would like to thank all the participants also to have come forward with all these questions and have stood with patience. I mean, this has been really an insightful talk also. And over to you, sir, for your remarks.
Yeah, thank you, Rakesh and Biprojit. I really enjoyed talking. And as I said, couple of questions like, we are almost sure they will definitely come. So you keep some answers ready. And there are many other things also. But as I said, my idea of interacting was to really do some kind of a thought provoking rather than only do a question answer kind of a situation. So some of my answers, which were on a broad-based part, is once again that's what I would like to once again come around to emphasizing. Come back to our analogy. Let's talk about cricket. So you have a bowling and batting. So similar, you have here the diagnostic and the therapeutic radiopharmacy. Then you have the manager, coach, and all that. So similar type here, the management of the radiopharmaceutical, nuclear medicine, marketing it, etc. They are all also equally important. Every player is important, right from Vinod Bankad or Polly Umrigar of the past to right now, whatever you can say as your Anil Kumble or Kapil Dev and so on and so forth, all the way down up to Virat Kohli and Akshar Patel and our, Ashwin. So everything had been like a process of evolution, importance-wise they are there. And they all have a very standard specific areas where they have contributed things like. And there is a case, there is a need for still marketing nuclear medicine per se. Once nuclear medicine per se is more marketed, then certainly, you'll have much greater opportunities to contribute some more of these, novel ideas or radiopharmaceuticals. So we are already doing well. We have grown very well in the 30, 40 years. Plenty of things have happened. 30 years back, there would not have been an MSc, IIT, nuclear medicine course there. They have all happened for the good and positive part evolution. So I presume it will continue, but we still have a job to do in many, many areas, engaging with the nuclear medicine to get more and more of our things into that standard of care kind of a situation. And then we would be in much greater business. And to that extent, all the younger people taking the field is extremely good. But please do not remain narrow focused. Radiopharmaceutical is a broad-based multidisciplinary science. Chemistry is important, one angle. Chemistry is not all like. And also instrumentation developments have been extraordinarily large. They have also taken place. We have to give equally due cognizance of that. And anything where we will have to agree that it's a closed chapter, we should not hesitate. Close the chapter, move on to something new. That what we call the sunset clause on some of the products or procedures will have to also be there. Keep that all in mind. All the very best to all of you. Thank you. I enjoyed joining you today. All the very best to you in the continuing session now. Thanks, Rakesh and Biprojit and all the participants. And any questions, anytime you have, you can always send me an email depending on my mood and timing. I may take some time, but I'll definitely respond. Thank you all. All the best to all of you.
Thank you, sir. Thank you a lot, sir.
Thank you, sir.
So we'll start with the next session from 4:50.